Welcome to our detailed exploration of the human microbiome in health and disease. In this chapter, we'll dive into the diverse microorganisms that colonize various body surfaces, the factors that shape their composition, and the implications of microbial disturbances on health. Let's begin with an overview of the human microbiome. A fetus develops in a sterile environment, but once born, it is exposed to a myriad of microorganisms. These microorganisms form complex microbial communities known as microbiota, which colonize the skin, nares, oral cavity, intestines, and genitourinary tract. These microbial communities play crucial metabolic and immunologic roles. The Human Microbiome Project, HMP, was launched in 2007 following the Human Genome Project. This 5-year initiative aimed to analyze the genetic composition of microbial populations in healthy adults. Samples were collected from the nose, mouth, skin, gut, and vagina. Through sequencing the 16S ribosomal RNA gene and whole genomes, researchers discovered significant inter-individual variation in species and genetic composition, with the gut microbiota showing the highest diversity. Despite individual differences, most people share a core microbiome. This core microbiome includes species present in 95% or more individuals at a specific site, representing a large portion of the microbial population. The secondary microbiome, consisting of numerous species in smaller numbers, contributes to individual variability. Functionally, the microbiome exhibits redundancy, meaning essential metabolic and immunologic functions are conserved despite taxonomic variation. The normal flora at each body site has evolved through symbiotic relationships with the host and competitive interactions among microbial species. Factors such as personal hygiene, diet, water sources, medications, and environmental toxins significantly influence the microbiota's composition. Changes in the microbiota can be benign if core functions are maintained, but some alterations can lead to disease by disrupting these essential functions. For instance, Figure 2.2 in the chapter provides an insightful example of the interaction between the microbiota and the immune system. This figure illustrates the various ways in which the microbiota influences and modulates the immune system. The gut microbiota, for instance, plays a pivotal role in the development and function of the host's immune system. It educates the immune cells, promoting the development of regulatory T cells that help maintain immune tolerance and prevent excessive inflammatory responses. Additionally, certain microbial metabolites, such as short-chain fatty acids produced by bacterial fermentation of dietary fibers, have anti-inflammatory effects and enhance the integrity of the gut barrier, protecting against pathogens. Dysbiosis, or alterations in the microbiome, can be indicative of various diseases. The chapter details several conditions linked to dysbiosis, including antibiotic-associated colitis, ulcerative colitis, obesity, type 2 diabetes, celiac disease, chronic wound infections, atopic dermatitis, and vaginitis. For example, overuse of antibiotics disrupts gut flora, leading to Clostridium difficile overgrowth, while altered gut microbiota can affect energy extraction and fat storage, contributing to obesity and type 2 diabetes. The relationship between dysbiosis and disease offers opportunities for advanced diagnostics and therapeutic interventions. Changes in microbiota diversity and composition could serve as biomarkers for diagnosing diseases. Therapeutic strategies to maintain or restore a healthy microbiome include probiotics, which are supplements containing beneficial bacteria that help restore the natural balance of the microbiome, prebiotics, which are nutrients that promote the growth of beneficial bacteria, and targeted antibiotics, which are designed to eliminate harmful bacteria while preserving beneficial microbiota. These approaches aim to correct dysbiosis and restore the microbiome to a healthy state. In addition, the chapter provides a glossary of terms that summarizes the most important concepts. Some examples already mentioned before and that you can review in this chart include microbiota, core microbiome, secondary microbiome, and other terms such as proteomics, metabolomics, and so on that you can take a look. In conclusion, this chapter provides a thorough examination of the human microbiome's critical role in health and disease. It highlights the importance of maintaining a balanced microbiome for overall well-being and the potential health consequences of its disruption. The insights gained from the HMP and other studies pave the way for innovative diagnostic and therapeutic approaches aimed at preserving or restoring a healthy microbiome. Thank you for joining us on this comprehensive exploration of the human microbiome in health and disease. Stay tuned for more detailed insights not only in microbiology, but also in various other branches of medicine. If you found this content useful, please like the video, share it with others, and subscribe to our channel. Don't forget to activate the notification bell to stay updated on all our latest content.